Mitosis and Meiosis by Jason Silva and Ramin Rahim. Mitosis is a division of the nucleus and genetic material passed on to two identical daughter cells. Mitosis is important for it enables multicellular adults to arise from a zygote along with the production of cells for growth, repair, and asexual reproduction. Interphase is when the cell begins a growth period. This period is divided into three different phases: G1, S phase, and G2. In G1, the cell increases in size along with the number of organelles. During S phase, DNA synthesis and replication occurs within the cell. During G2, materials for the next mitotic division are repaired. These three stages of growth make up most of the cell cycle, with only a small part being mitosis cytokinesis. During interphase, the cell is yet to divide, and the chromatin are enclosed in a nuclear envelope. Outside the nucleus, two centrosomes lie adjacent to one another. During prophase, the nuclei disappears while the chromatin begins to condense into chromosomes. By the end of prophase, the centrosomes have migrated to opposite poles, thus forming a mitotic spindle, as the newly formed chromosomes move towards the center of the cell. As the centrosomes move to opposite poles, they form microtubules, which connect to the kinetochores of the chromosomes. These microtubules are responsible for pulling the chromosomes apart and are named kinetochore microtubule. During metaphase, the chromosomes line up across the metaphase plate halfway between the two centrosomes. After the chromosomes have lined up evenly, the cell is now in anaphase, where the chromosomes separate at the centromere, leaving two sister chromatids. The new chromatids are now their own chromosome after separation. Telophase and cytokinesis are the last phases of the cell cycle in mitosis. During telophase, corresponding sister chromosomes attach at opposite ends of the cell. A new nuclear envelope forms around each of the separated sister chromosomes. Both sets of new chromosomes unfold back into chromatid. Cytokinesis is where the division of cytoplasm occurs, thus forming two new cells. In animal cells, a cleavage furrow is formed, and in plant cells, a cell plate is formed. This process creates two new identical daughter cells, each diploid with the same number of chromosomes as the parent cell.
Meiosis is a modified type of cell division found in sexually reproducing animals. It is important because it is used to produce genetically varied haploid gametes. This variation can be essential to the survival of a species. Meiosis is divided into meiosis I and meiosis II. During prophase I, chromosomes condense and the nuclear envelope breaks down. Replicated homologous chromosomes pair up through a process called synapsis. During synapsis, crossing over or the exchange of genetic material occurs between non-sister homologous chromatids, forming sites of chiasmata. During metaphase I, the homologous chromosomes line up along the metaphase I plate. The chromatids in each homologous pair are attached to the kinetochore microtubules from each pole of the cell. During anaphase I, homologous chromosomes separate and move towards opposite poles, guided by the spindle. Once the sister chromatids are on opposite ends of the cell, telophase I and cytokinesis occur. During these stages, the cell divides to form two daughter cells. Now the cell is in meiosis II. Note that no replication of the chromosomes occurs between meiosis I and meiosis II. In prophase II, a spindle forms, and the chromosomes, which are composed of two chromatids, move towards the metaphase II plate. In metaphase II, the sister chromatids are lined up along the metaphase II plate. Note that the chromatids are not genetically identical. The kinetochores are attached to the microtubules from opposite sides of the cell. In anaphase II, the chromatids move towards opposite poles as individual chromosomes. There is a breakdown of the proteins holding sister chromatids together at the centromere. During telophase II and cytokinesis, the chromosomes begin decondensing, the nuclei form, and the cell divides. With this division, meiosis is complete. A total of 4 haploid cells that are genetically distinct from the parent cell have been created. In addition, each cell has half the number of chromosomes as the parent cell. Although meiosis I and II are similar, there are some key differences between them. In meiosis I, diploid homologous pairs form, and synapsis occurs. Then the homologous pairs separate, resulting in 2 haploid daughter cells composed of replicated chromosomes, or sister chromatids. In meiosis II, haploid sister chromatids split, resulting in 4 haploid cells with unreplicated chromosomes.
Mitosis and meiosis have some key differences. In mitosis, DNA replication occurs during interphase before mitosis. There's one division and the result is 2 diploid cells, genetically identical to the parent. In meiosis, DNA replication occurs during interphase, before meiosis I. There are 2 divisions. In addition, synapsis occurs and results in 4 genetically different haploid cells that contain half the number of chromosomes as the parent.